Hello and welcome to our symposium. This event has been in the works for a long time, and I am so honored and proud to have assembled a group of amazing ichthyologists. I'm Liz Miller, and I'm the chair of the symposium along with Luke Tornabene and Prasanto Chakrabarty. In the first half of this talk, I'm going to introduce the goals of the symposium, and in the second half, I'm going to share my take on why there are so many kinds of fishes. As a kid, I remember knowing what I wanted to do with my life by the age of 3. I really wanted to be a herpetologist. And it wasn't until I took an undergraduate ichthyology class that it planted the seed that, hey, fishes are really cool too. Especially interested in biodiversity and macroevolution, there's no way to get bored working on fishes because there are so many kinds of fishes. In fact, there are over 35,000 kinds of fishes. And the number I just gave you is the sum of a whole bunch of parts, and species richness is not evenly distributed among groups of fishes. And so for many people, it's important to also answer the question, why are there so many kinds of cichlids, or gobies, or coral reef fishes, or tropical fishes? So my principal interest is in species richness, which means I treat one species about the same as any other species. And the way I study fishes usually is through rows of an Excel spreadsheet. And for a lot of people, that's a really boring way to study fishes, because there's a huge amount of morphological and ecological diversity of fishes. And it's really fun to argue with other ichthyologists about what the weirdest fish is. So the fish that's like right above my head here is the only fish that can bend its neck. So having a neck is pretty weird. But if you think you know a weirder fish that I haven't put on this slide, I really want to know what it is, so please share it with me. So this is why we've invited speakers that think about this problem in really different ways. And when it comes to answering this question, I look around and I see early career researchers at the forefront of this question. And here are just a handful of examples of papers that have come out in the past few years in which early career researchers made massive contributions to. And these papers are making the rest of our jobs a lot easier because they're building big data sets, or they're inventing new ways to analyze those data sets that are going to be used for the next 5 or 10 years. So I think part of the reason for the success of early career ichthyologists is because we're really diverse, both in our scientific perspectives, but also who we are as people. And our diverse perspectives as people kind of shape our diverse perspectives as scientists. And to demonstrate this, I'm going to show you demographics of the ASAH membership. So we have responses for about 300 professional ichthyologists, about 1/3 of which are students or postdocs. Note that pre-tenure faculty are also considered early career, but the survey didn't distinguish tenure status, so they're included in the permanently employed group. So you can see among early career ASAH members, we have more diversity in race, in gender, sexual orientation, and in these other measures of diversity. And this is really exciting because it means that the future leadership of ASAH is potentially going to be way more diverse than it ever has been. But what will derail that future is if those early career members are more likely to leave the society if they're not members of privileged groups such as white or male or other groups that have some sort of privilege. And once someone finds a society where they feel like they belong, they will be members for their whole career. So the ASAH is competing with other societies for this leadership and talent. And if we lose early career members, we risk losing on leadership, innovation, and we risk the financial health of our society. So it is essential to the future of ASAH that we do all we can to retain early career members, especially those that are not members of privileged groups. So the symposium has a dual purpose. First is to answer the question, why are there so many kinds of fishes? But also to showcase the early career scientists that are working to answer that question. Early career researchers are a huge portion of ASAH membership. And I believe members don't belong to a society, but rather the society belongs to its members. and this is our society too. This symposium is going to demonstrate that early career researchers are leaders and innovators, and we are the future leadership of ASIH. With that, I'll transition to sharing my own take on the symposium topic. The goal of my research program is to explain why species richness varies. Ravenfishes live broadly across the world in all aquatic habitats, so they're perfect for this type of question. Today I'm going to share 2 short vignettes from my work that I think show some important lessons as we move forward to explaining the diversity of fishes. Speciation is the process that generates new species, and rates of speciation vary across clades. And that speciation rate variation may be linked to organismal traits or external factors like environmental change. However, what if factors that influence speciation matter at small scales but then don't scale up to explain broader levels of variation? So sexual dichromatism is one factor that's been established to matter for reproductive isolation and speciation. This has been hypothesized since Darwin, and it's been shown independently in all these fish groups. And it's a potential driver also for the adaptive variation of African rift-lake cichlids. So if this is a process that scales up, we might expect the presence or absence of sexual dichromatism in certain groups to help explain diversification rates across fishes, right? And the short answer is no. We collected this information across ravenfish species and compared diversification rates at the broadest possible scale among all ravenfishes. And here we see no difference in diversification rates between monochromatic or dichromatic fishes. So then I wondered, what if individual groups do show a relationship but maybe in different directions, and when you put them all together they swamp each other out? So I delimited 18 clades of fishes based on orders or families and fit diversification rate models on these clades individually. So the pie chart shows the proportion of model weight of trait-dependent models of diversification versus trait-independent models of diversification. So clades with more dark gray have stronger support for a role of sexual dichromatism on diversification. And we see that, a few clades do show a relationship, but where are the cichlids? They're over in the pile of light gray, which is the majority of clades. They still aren't showing this relationship. So what the heck? If anything, we should see it in cichlids, right? So to investigate this further, we didn't use ranks, and instead we sliced the tree at these particular time cutoffs. And the clades that resulted in that slicing, we fit models on those clades individually. And the pie I'm showing you is just for cichlids. So we don't see anything at 100 or 80 million years. And we still don't see anything at 60 million years. And by now, the time slice has cut Cichlidae into 2 groups. But yes, okay, we see the relationship at 40 million years. Independently in these 2 groups of cichlids, there is a role of sexual dichromatism. And this is nice because it's what we expect, but there's a couple things that are disconcerting about this. First of all, people like to study ranks. Your paper is about Cichlidae, not this random clade that's based off of a time cutoff. And the role of trait-dependent diversification isn't necessarily predictable at the level of certain ranks. And the second disconcerting thing is that we put a lot of effort into understanding speciation at really small scales, ecological scales, and it doesn't seem like the same traits that we look at small scales are those that matter for this really broad variation. And that certainly does not mean that those traits are irrelevant for speciation, just that there may be other traits that are stronger at controlling this among clade broad scale variation. So we've talked now a lot about speciation rates, and we're doing that with the assumption that speciation rates are related to species richness. And that's a very fair assumption. You have species, speciation rates, species richness. It's in the names. However, we're coming to terms with the possibility that speciation rate variation is just one possible explanation for species richness patterns, and it may not even be a very common explanation. And this is shown very cogently by the inverse latitudinal gradient in speciation rates. Where species richness is highest in the tropics, speciation rates are highest in the poles. And I've worked on similar questions in different systems, and I really believe that this is a general pattern, where the habitat or place with the most species is not the one with the fastest speciation. Today I'm going to show you an example comparing freshwater habitats, but if you like these other systems, check out these other papers. So one of the most intriguing species richness questions is that half of fish species are freshwater and the other half are marine. And the reason why that's weird is because less than 1% of Earth's water volume is freshwater. And so you picture a river, you think of fragmentation, which will isolate populations. And so it's hypothesized that freshwater habitats promote isolation, which promotes speciation, so freshwater diversification rates should be faster, allowing them to catch up with the ocean. This hypothesis is intuitive, but the evidence for it is actually equivocal. Freshwater diversification rates are slightly higher, however, there's still a lot of overlap. Freshwater is a pretty broad category, so to investigate further, I broke freshwater up into different habitat categories. So we see that most freshwater species live in rivers. However, diversification rates are way faster in lakes, even though lakes are a small fraction of freshwater diversity. And what's perhaps even more surprising is that rates in rivers are similar to that in the ocean. So the idea of lots of speciation in freshwater because of this fragmentation isn't really matching what we're seeing here. So if not speciation rates, then what explains the freshwater paradox? Here I'm showing you an ancestral state reconstruction of habitat on Teleos, and you can see that fishes have been diversifying in rivers and in the ocean for similar amounts of time, about 100 million years. So if you're diversifying at similar rates for a similar amount of time, it's suddenly less surprising that you end up with similar richness at the end. So it's not necessarily that freshwater has all this fast speciation, more so that it has constant speciation over this long period of time. So today I've shown you some ways that our first instinct about evolution can be led astray. But I'm not feeling pessimistic, I'm feeling very optimistic. It's a wonderful time to be an evolutionary biologist because we know more about the history of fishes now than we ever had. And that's because of all of our efforts building phylogenies and understanding the fossil record and inventing new ways to analyze that data. And I think you'll share my optimism after seeing our speakers today and their own takes on why there are so many kinds of fishes. Thank you.